So first, I want to just thank the PacBio Prism Roadshow folks for inviting me here to be able to present on our latest developments in fusion transcript detection. So RNA-seq and transcriptome sequencing in general is really a powerful method for getting insights into cancer biology and studying cancer samples. It provides us with both genetic readouts and functional readouts. All we need to be able to do is resect the tumor sample, convert the RNA to cDNA, and then you can sequence it using whatever your favorite modern sequencing technology is. And once we have those reads, then we can get insights into those functional and genetic readouts. From functional readouts, we have expression information that we can get. We can study the individual isoforms that are sequenced, get insights into chimeric transcripts, which might represent fusion transcripts, and RNA editing, insights into single cell resolution. And from the genetic readouts, we can read out polymorphism data, mutations, insights into structural variations, and copy number variations or viruses in the sample. There's so much that you can do with transcriptome data. And you really need a powerful set of tools to be able to extract the most from these data. And we've been developing a toolkit called the Cancer Transcriptome Analysis Toolkit. CTAT is short for that. And we have various modules that we've developed to focus on transcript reconstruction, pulling variants from the reads, fusion transcripts, of course, alternative splicing, viruses, and getting insights at single cell resolution, looking at tumor heterogeneity, such as copy number variation. Much of the work that we've done over the last few years is really focused on developing modules for studying fusion transcripts, with a big focus on the Illumina RNA-seq. And we have a few different tools that we developed for that. We have Star Fusion, we have Trinity Fusion, and we have Fusion Inspector. And most recently, we've added a long read support for fusion transcriptome, for fusion transcript detection in our new tool called CTAT-LR-fusion that I'll tell you more about today. So identifying fusion transcripts is really important for clinical studies. There's many fusion transcripts. They're actually the primary causes of cancer, the primary drivers of cancer. And these fusion transcripts tend to occur through just genome rearrangements, translocations. One of the best examples of that is this one, which involves a translocation between the bottom arms of chromosome 9 and chromosome 22, which brings the BCR and ABL1 genes together, positions BCR 5' to ABL1 3', generating the BCR ABL1 fusion transcript and fusion protein. And this is a driver of cancer in 95% of CML, chronic myelogenous leukemia patients. Now, you can detect this fusion event from whole genome sequencing. Of course, that gets very expensive. It's much cheaper to do it using transcriptome sequencing, and transcriptome sequencing gets you right to the functional events that are involved in cancer. So it's very attractive to be able to detect these from just transcriptome data alone. There are many such fusion transcripts that are relevant to cancer biology. BCR-ABL1 is just one example where the most of the cases is the hallmark fusion event, but there are over 300 different fusion transcripts that have been identified that are relevant to cancer biology. You'll find them in the COSMIC database, and some of these are true hallmarks of the disease. For example, in synovial sarcoma, you have this SS18 SSX fusion, which is in 100% of the cases. And even in prostate cancer, 50% of the cases are due to TMPRSS2 ERG fusion. So it's important to be able to identify these. And also, from a treatment perspective, it's important to identify these because some of these involve kinases. And these kinase fusions can often be treated with kinase inhibitors, leading to favorable patient outcomes. Now, detecting these fusion transcripts from RNA-seq data, particularly in the earlier days, using Illumina RNA-seq, involves a couple of different strategies, a couple of popular strategies. And one, we can just take the short reads and align them to the genome and look for split reads or paired ends that align to different genes and suggest that there's a fusion event. Or we can take those short reads and we can assemble them into longer transcript sequences, align those transcript sequences, those assembled transcript sequences to the genome, and look for those that are chimeric transcripts, suggesting that there's a fusion event. So we have these two different major paradigms and be able to search these events out. And we develop tools that basically use these strategies as the primary mechanisms. We have a tool we developed, the Star Fusion, which searches for chimeric read alignments. And we have Trinity Fusion that we developed for first doing assembly using our Trinity assembler, and then looking for chimeric fusion transcripts. Most recently, we developed a tool called Fusion Inspector, which performs analysis of the sequence context and the expression of the fusion transcripts, models the fusion transcripts as fusion contigs, realigns reads to those fusion contigs in order to perform quantification analysis of breakpoints. We have visualization tools that are also included here to really highlight the actual read alignment evidence that supports these different fusions that we're finding. Now we're in the era of long reads, and there's been massive progress, of course, over the last decade in development of long read sequencing, especially for transcriptomics. Both these major technologies, higher throughput, lower sequencing error, and the latest information on sequencing error rates is just incredibly impressive. And there's been a major development recently, resulting in what's called MassSeq by Aziz Al-Khafaji, who's someone that I work with at the Broad Institute. He developed this method that increases the throughput of PacBio sequencing for transcriptomic reads, giving you anywhere from 8 times to 16 times as many reads per flow cell. So now we can routinely get 40 to 120 million cDNA reads from a single flow cell. We do this for bulk transcriptomes, and we can do this for single cell transcriptomes. So very powerful, and this is the approach that we really want to be able to leverage for fusion transcript detection, both in bulk samples and in single cell samples, leveraging these long reads that we get from the PacBio. What's actually commercialized now is Kinex. We buy it off the shelf. And we want to be able to adapt some of the tools we developed earlier for short reads to get them to work with longer read sequences. And Trinity Fusion seemed like a good candidate, given that it would assemble transcripts into short reads into longer transcripts. So instead of doing any assembly here, we can just take the long reads themselves and fit them into this process to find examples of chimeric long reads. And for Fusion Inspector, we can use modules for modeling the fusions that we identify, realigning those long reads to these fusion contigs to identify breakpoints and characterize the expression of those different fusion isoforms, and also use the visualization components that we have from this toolkit as well. So we did that. We came up with what we call CTAT-LR-fusion. CTAT-LR-fusion has a couple of different steps. In the first step, we take the reads and we quickly identify those long reads that look to be chimeric, and that part of the read is aligned to one gene, and another part of the read is aligned to another gene. We use this using a customized version of Minimap2 software called CTAT Minimap2, which is 4 times faster than the regular Minimap because it's just focusing on getting alignments for those reads that look like they're chimeric and that they're mapping in multiple locations instead of trying to get best alignments for all reads. And once we have that list of candidates, then we use the Fusion Inspector-like approach for characterizing them. We model the fusion contigs, we align those reads identified as candidates to these contigs, and we identify the fusion transcript breakpoints and expression data and provide the downstream visualizations. So there are other tools that operate in this space. There's a handful of tools that we have here, and we want to be able to compare our approach to the other tools that exist in this area. We use a couple different data sets for doing this. We're doing benchmarking. We use simulated long reads, and we use genuine long isoform reads. For simulated reads, we simulated both PacBio, Hi-Fi, and ONT reads using a tool called PBSim3. And for the genuine long reads, we have a few different data sets that we use. We have a Sericare fusion RNA standard that I'll tell you more about. There's 9 tumor cell lines that we sequenced in collaboration with the DepMap project at the Broad. And we also focus on tumor single cell transcriptomes. There's a couple of different data sets here, a couple of cancer data sets that we had previously published or collaborators have published on that we could use for searching out fusion transcripts. So we leveraged all of these. I'll first tell you about the simulated data. Our strategy for doing the actual benchmarking or accuracy computations involves having a truth set where we score the number of true positives and the number of false positives as a function of the minimum read evidence support we have for those fusions. From those true positive and false positive counts, we can get our precision and recall statistics. And then based on precision and recall, we can get our F1 overall summary accuracy score. So we can plot this accuracy score, the F1 metric, as a function also of minimum read support. We'll often find a peak accuracy, and we can use that for making comparisons among these different tools. We have when we did the benchmarking, we did it at variable levels of stringency. First, you could just score a fusion as just finding that gene pair, regardless of the breakpoint, regardless of the orientation of those gene symbols. Just do you have a candidate that has that pair of gene symbols? We can score that, or it can be really strict and say you have to have that pair of gene symbols and they have to be in exactly the same order as you expect for that fusion transcript. So we can be really strict, or we can be lenient and allow for either orientation. We can also look at the breakpoint resolution. So do we have actual exact breakpoints for the fusion transcripts that we're detecting, or do we need to allow for fuzzy breakpoint, allowing a little bit of space around the breakpoint in order to identify it as a true positive in these different predictions? So we have these different ways of doing very strict or very lenient scoring when comparing these different methods. And I'll just show you first the results from our simulated experiment. Here we have the simulated PacBio reads. We did 5 samples with 500 fusions in each sample, and here's the results according to the different levels of different ways in which we compared them, requiring strict gene symbol ordering, allowing for either orientation, requiring strict breakpoint resolution, or allowing for fuzzy breakpoints. And curiously enough, it's really only our tool, CTAT-LR-fusion, and Jaffa-L that accurately provided both fusion reporting with requiring strict ordering of the genes, so correct ordering of the genes, and also having exact breakpoints defined. We found very similar results if we did the simulations with the ONT data. So really, it's our tool and Jaffa-L that were performing best with the simulated data. And I just want to stress the importance of getting the ordering of the genes right. We're not just looking at structural variations here, where you just know that if you have a structural variation, you have translocation between 2 chromosomes. When you talk about fusions and fusion transcripts, the gene ordering really matters. It's the difference between having cancer in some cases and not having cancer. Just to give you an example, here we have TAC3 and we have FGFR3, which are neighboring in the genome. There's only about 100 kb or so that separates the two. Now, if we found a fusion transcript that involved TAC3 at the 5' end and FGFR3 at the 3' end, then you might imagine this is just read-through transcription and not really think too much about it. Read-through transcription happens a lot. There's a lot of examples of that. We don't necessarily know the full relevance of that, but that construct has not been shown to be relevant in cancer yet. But if we found the other orientation where FGFR3 is at the 5' end and TAC3 is at the 3' end, well, this would require a somatic rearrangement that repositions the FGFR3 gene, putting it 5' to TAC3. This causes cancer, all right? So there's a big difference, and it's really important to get the gene ordering right. Other data sets that we use for benchmarking, including like real data now instead of the simulated data, involve a few different data sets. One is the CTAT-LR-fusion standard. So this is just it's a kit that you can use for fusion validation assays. You can buy it off the shelf from CTAT. It's a cell line RNA background that has introduced into it 16 different fusion transcript constructs. So we can basically score with our tools if we sequence these using short reads or sequence it using long reads using Kinnex. Are we able to find them with our tool and the other tools? Here's the results from sequencing these data using Kinnex and also using Illumina short reads. We sequenced 6 to 10 million reads of Illumina and 1.5 to 3 million reads each for the PacBio. We actually had, in terms of sequence bases, we had more sequence bases from PacBio than we did Illumina. So we actually downsampled the PacBio to reach the same number of sequence base pairs to make this more equivalent kind of comparison. And what we find is that in the case of the long reads, right, the isoseq, we're finding each of these fusions, and we find them at least like 10 to 30 reads per sample. In the case of the Illumina data, we also find all the fusions across all the samples, but we did these in triplicates, right? In some of the triplicates, we actually don't find any reads from the short reads, right? But we do find it with the long reads. So in this case, the long reads were much more effective than the short reads at the same sequencing depth. Now, if we compare this is based on our CTAT-LR-fusion results and using Fusion Inspector, which is our short read tool for finding fusions here. If we compare CTAT-LR-fusion with the long reads to the other tools that are available for long reads, we find that we find all these fusions in the CTAT sample, and PB Fusion also found the latest version of PB Fusion also found all the fusions, but the other tools failed to find at least one of these fusions, and that was consistent across replicates. Another data set that we use to explore fusion detection is cell line transcriptomes, cancer cell line transcriptomes, and done in collaboration with the DepMap project at the Broad. We selected 9 different cell lines where we sequenced the transcriptomes, again, using long reads and using short reads. And these 9 cell lines were selected because they represented a diverse set of different cancer types, including breast, prostate, lung, and blood cancer. Each of them harbors a known fusion that's relevant to that cancer biology, and the fusions are listed here. So we sequenced this using Kinnex, And we explored all these different tools for being able to find these different fusions. So here we have the 9 different fusions, and we have the number of reads uh, identified as supporting the fusion by each of these different tools. You'll see that some cell lines, like VCAP, which is a prostate cell line, prostate cancer cell line, has a lot of fusions found by each of these methods. Others, like MJ, has very, very few. Most of the tools predicted similar numbers of fusions across these different cell lines, except for Fusion Seeker, which tended to overpredict across the board. And the the predictions that we have across these different programs are relatively consistent too. So if we look at the minimum number of fusions where at least 2 predictions agreed, that's shown here in this black line. So you can see that they're pretty consistent. If we look at just those oncogenic fusions that we knew that these cell lines harbored, and were they found by these different tools? Yes, they were all found by all the different tools. And the number of reads that were found to support them were fairly consistent across the board, anywhere from very few number of reads, like only 2 reads here in this BCR-ABL1 fusion, to almost 500 reads in one of the other fusions. Now we want to be able to use this data set for benchmarking as well. So how do we do that? This is real data, not simulated data. We don't necessarily know exactly like a perfect truth set for these data. So in this case, we make a proxy truth set. We identify those fusions that are supported by at least 2 programs, which is what we have here. 133 fusions were supported by at least 2 programs. And there was a small number of additional fusions where we had both long read and short read support, but they're uniquely predicted. We included those in our truth set. It was 145 proxy truth fusions. Using this for benchmarking, and here we're looking at the accuracy of these different programs according to the minimum number of long reads that we had, and our program had the highest F1 score, overall summary accuracy, across the board. So we're doing quite well in comparison to these other programs. Some other interesting things that were coming out of sequencing the transcriptomes of these DepMap cell lines. There's some fusions that actually had evidence for multiple splicing isoforms. This is just one example, and these multiple splicing isoforms were supported by both short reads and long reads. Here's a case where we had the CYTH1 EIF3H fusion in the breast cancer cell line, and the first exon of CYTH1 you can find spliced into 5 different exons in the EIF3H downstream, and this is supported by both long reads and short reads. The expression values that we get for these fusions are very consistent between fusion reads. Between the short reads and the long reads. You can see we have a near perfect correlation of read support for each of these different breakpoints for long reads versus short reads. And this is just another view where we have the gene structures here plotted. So you can see the actual position of the breakpoint and the relative evidence that we have for supporting each of these different breakpoints. This one's kind of curious because you actually have a decrease in read support as you start going downstream in the 3' gene. Not all the fusions that have multiple isoforms are exactly this clean. You can see we have this really nice clean one. The others are not necessarily as clean. There are some places where they agree with short reads and long reads, other places where they disagree. There are places where the short reads, the dominant isoform supported by the short reads is not the same as the isoform supported by the long reads. And there are cases where we actually find additional fusion isoforms from short reads or long reads, but not the other. So just a couple examples here. This is the Tempest 2 ERG, where we have long read support, we have short read support, and you can see that there's different breakpoints. There's a case here where we have it from long reads, but we don't have the fusion found from short reads. And in this other fusion, TATDN1 GSDMV, there's many fusions. I think there's like 13 to 15 different isoforms for this one fusion pair. And there are many cases where we actually have short read evidence, but we actually don't have long read evidence. So it can go either way. And CTAT-LR-fusion incorporates the fusion inspector. So if you have short reads and you have long reads, you can integrate them together in order to maximize your sensitivity for these things. Now, for single cell data, there's a lot of interest in being able to detect fusions at single cell resolution to gain greater insights into tumor heterogeneity. And the 10x approach, 10x genomics chromium, is one of the most popular ways to do this. And with 10x, we have beads, or they call them gems, that have barcodes attached, or oligos that capture oligos, capture the RNAs from the 3' end from the poly A tails. And once you have your RNAs captured on the gems, you've done your library preparation. You can sequence it on either Illumina, or you can sequence it on the Revio. You basically split it up and do like half and half if you want to, which is really powerful to get both. And then, so we basically, this is the approach that we would use for gaining insights into fusion transcripts at single cell resolution. There's a couple of different projects that we looked at in order to examine this. One is this melanoma sample where you have T cell enriched melanoma sample, and this was published last year as part of the original MassSeq paper. Here we have millions of PacBio isoseq reads. We have 200 million Illumina 10x 3' reads, about 7,000 total cells. And here we have cancer cells, and we have normal T cells. And we assayed these cells for fusion transcript detection using our tools. We use CTAT-LR-fusion for long reads, and we used Fusion Inspector and Star Fusion for short reads. And what we basically found is there was one fusion identified in the sample, this NUT M2A AS1 fusion that was found enriched within tumor cells, and there's very little evidence for finding it. There's only a few cells that showed up with short read evidence, and it's probably from ambient RNA, so they're probably not even there. But all the long reads showed up in the tumor cells, and most of the cells that we actually detected having fusions were from long reads. You can see most of the cells were basically long reads. If we combine with the short reads, we can get greater sensitivity for cells. But if you want to find fusions in tumor or single cell data, you kind of need these long reads. You kind of need the single cell Kinnex kit in order to be able to do it. Other advantages to using the long reads in this approach is you actually get full length reads that correspond to these fusions. In this case, with this one fusion, there's actually 8 different isoforms that we find from the long read data. And the long reads that we find are almost entirely full length across the board. If we look at the structures that are supported by the Illumina reads, we only basically find evidence for just the breakpoints. And there's a handful of isoforms that we find evidence for, but it doesn't give you the full structural context, right, as you do with the longer reads. Another data set is corresponds to ovarian cancer, and this is with our collaborators. And here it's just one patient sample. We actually looked at a few different patient samples. We have 54 million PacBio isoseq reads, 35 million Illumina, about 500 cells. There's 4 different cancer-specific fusions we detected. These are all the cells from that sample. We've highlighted the cancer cells, and there's a couple fusions here that we highlight. And what's interesting here is that the cancer cells look like they form 2 different clusters, and we find different fusions in each of these clusters, suggesting that there's tumor heterogeneity. So we pull out just those tumor cells and we analyze them separately. This confirms that we actually do have 2 clusters of cancer cells indicating tumor heterogeneity, and we do find that there are fusions that are separately enriched. Here I'm highlighting all 4 of the fusions that we found. And another interesting thing here is that 3 out of the 4 fusions that we found only found them using long reads. So overall, to summarize, fusion gene order and orientation is really critical when you're reporting the fusion transcripts, because if you get the orientation wrong, it's a different fusion entirely. May cause cancer, may not cause cancer. Got to get it right. We showed here that CTAT-LR-fusion, our new tool, really excels at long read fusion detection. We showed this with simulated reads. We sequenced a couple samples using Kinnex, both DepMap samples and the fusion standard mix. In both cases, we demonstrated that we had superior performance. And there's other key merits of these long reads. You get many more cells that are going to be detected with fusion reads. And again, if you're doing single cell transcriptomics and you want to find fusions, you really kind of need this. And the full-length fusion isoforms we're detecting is really another great merit, as opposed to these partial splice junctions that you get from the short reads. So super helpful. If you're interested in more in our software, it's on GitHub. It's freely available. It's open source. It's really easy to use. It gives you nice interactive visualizations that are IGV based. We have a preprint that basically describes everything that I just showed you now, but in more detail. So if you're interested in learning more, talk to me, read our paper. And these are all the folks that are involved in this effort. We have our our methods development lab, which is led by Aziz Al-Kafaji, who leads this group that I'm part of. And then we have the DepMap collaborators here. Victoria Popik is another PI at the Broad that's been helping out in this work, and our Swiss collaborators that provided access to and helped us analyze the ovarian cancer samples. So that's pretty much it. I'm happy to take questions.